Hello everyone and welcome to today's live broadcast, DNA Standards and Bioinformatics Benchmarks for Metagenomics, presented by Dr. Christopher Mason. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon on the lower right-hand corner of the slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button in the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Christopher Mason, Associate Professor at Weill Cornell Medicine Department of Physiology and Biophysics. Dr. Mason's laboratory builds and deploys biochemical and computational technologies to understand and engineer human and other genomes. He is a principal investigator or co-investigator on several NASA missions and projects. Some missions will discern the molecular basis of changes in the human body during long-term human space travel, including genetic, epigenetic, transcriptional, and metagenomic changes. One additional mission is optimizing a new method of space-based DNA nanopore sequencing, and his group is also creating other genetic technologies and algorithms that can measure nucleic acids in space. Notably, the nanopore-based sequencing methods he has pioneered with NASA and UCSF scientists will enable mid-mission rapid pathogen diagnostics for astronauts, create new applications of sequencing for space exploration, and discover novel DNA and RNA-based modifications. I will now turn the presentation over to Dr. Mason.
Great, thanks so much for the introduction. Pleasure to be here today. I'm going to go through a number of points about what is the current state of the art in genetics and metagenomics in terms of our standards that we're using to better develop new tools as well as algorithms to understand the world in us and also the world around us. So I'll start first with, let me pull up here, okay. So first is a little bit of history. If you think back of what used to be the case for just simple things like threads on screws and what was the distance between them, people literally couldn't build things or even exchange different nails and screws and fundamental bolts that they would need to build things for houses, for businesses, for anything that you would want to put together. And so the standards that were created over the past 200 years make it such that you can go to any hardware store today and go back to your house and build anything or repair it or put it back together and tinker with it a bit. And so this is many years coming, but in sequencing space and in genomics in general, there was historically a lot of work on microarrays that created a lot of confusion about what was the best microarray, the best way to, in a high throughput fashion, measure thousands or even these days millions of loci simultaneously. This was true up until the early 2000s, or I like to say is the early noughties, the 000 days of the 2000s. But what ended up happening after the sequencers, a large proliferation of many kinds of sequencers in 2005, 6, and 7, is that suddenly there was a similar problem where there's a proliferation of methods for doing sequencing, but no real standards that had yet been developed. And so one of the calls to action from Nature Biotechnology in 2008 was to really try and develop something like the MAQC, or the Microarray Quality Control Consortium, to really set these standards up so people could really understand what they're looking at and actually understand the algorithms that could have their pros and cons for the measurement of things that you do sequence. And so this led to a lot of international collaborative work between industry, academics, and government scientists, what was called the FDA Sequencing Quality Control Consortium, as well as the ABRF Next-Gen Sequencing Study, and led to a series of manuscripts in 2014 that really did a lot of deep benchmarking in RNA sequencing. And so you can see here, this was a lot of the work that we had done with many colleagues and put out here on the existing platforms. But what's interesting, well, there's a number of interesting things about the study, but one of my favorite things about the study was when we'd sequenced even some samples out to billions and billions of reads, we kept finding that there was no real plateau, as you can see on the right. We did see that the number of reads mapped to different annotations would depend to some degree on your annotation itself, and so the genome that you will see in the transcriptome that you will measure is to some degree, and even a large degree in some instances, dependent on your annotation. But really, it means that as you keep sequencing deeper and deeper and deeper, this question of how many rare isoforms and transcripts could there be, it means we kept finding more and more isoforms, more and more genes present at very, very low levels. And so we think that this means there is no real complete, basically ceiling. It seems like we kept finding, partly due to cellular heterogeneity and complexity and rare alleles present. But you can see here that we did keep finding things. And so what's interesting is that today, when you do metagenomics research, we can actually see that when you take the same sample as you can look at here and you sequence out to hundreds of millions of reads, you keep detecting new species. They keep kind of popping up using, say, a metaplan in this instance, to discover new organisms that are present. And so what you do at 5 million, 10 million, or 30 million reads, the problem there is that you'll keep finding new organisms. So this leads to a question of, well, how many actually are there in my sample? How many total organisms can I expect to find? And this is a relatively unknown of an answer in metagenomics or even in microbiome research. And so we've been working with a variety of groups to look at this. And most notably, one of them is also, again, the NIST Genomes in a Bottle Consortium, as well as the Sequencing Quality Control Consortium Phase 2, which is the SEXI2 study from the FDA, and also the ABRF NGS Phase 2 project, which is looking at these questions. And we're releasing all these data online as they get QC'd and finished, with the goal to have a lot of them finished in the next couple months. So one interesting thing is, if you take the human genome as a perspective, we can see here that doing 7 human genomes with deep characterization and posting all the data online has led to a lot of people being able to grab the data, compare how they're doing in their lab, relative to these sort of clear international standards. This is also in concert with the FDA and Precision FDA, so that as you, again, generate a new protocol, new method, new sequencing, basically protocol in your lab, you can immediately compare to how you're doing relative to these international standards. The other thing that we've seen is, there's no standards for other kinds of areas. So, for example, there's no standards yet for a strange pink lake that exists. For example, here you can see this Lake Hillier from Australia, and there's no standard for a weird, funky halophilic pink lake. There's no standards yet for example, the Gowanus Canal, which is a Superfund site. You can see here that this is essentially a complete toxic sludge site that we're taking samples from. But how do you standardize extraction or methods from something like this? There just really is no good standard. So we want to have standards applied to all these areas. Or this, there's no standard for a strange collection of an organism that you might find on a subway surface or that people are clearly afraid to touch. And so there are some standards that have been begun, but historically works like from the MBQC, the Microbiome Quality Control Consortium, did do some extraordinary pioneering work in this direction, but a lot of it was initially focused on 16S, and sort of the MBQC Phase 2 is going to do a little bit of both 16S and shotgun, and look at extraction and storage and all the sort of complications and caveats of different methods in metagenomics and microbiome research. And so it's an exciting time because some of these open questions are now beginning to be addressed. And so I'll discuss some of them today and some of the preliminary data we've seen. There's also a number of other consortium groups, the microbiomestandards.org group, which is part of IHM. There is also even a lot of work at the NIH to make skin microbiome standards, which is relevant to very particular skin research groups. And finally, there is some work at NIST. There was a workshop just last fall on standards in microbiome measurements, which has led to an international standards group called IMMSA, the International Metagenomics and Microbiome Standards Alliance, which again is a collection of many people in industry, government, and academia all working together. So one thing I want to point out is when we think about what you're measuring, I'm going to switch gears a little bit to what's in the organism and when you measure what it is. We can see here that the 16S RNA gene, for example, which has been historically broadly used for metagenomics research, is not actually even the best gene to use if you had to use a gene to separate distantly related species, and most especially closely related species. You can see here in green, these are 74 different marker genes that are present in most prokaryotes. And 16S ribosome RNA is not even the best gene for either of these categories, distantly or closely related ones. It's just one that historically has had a lot of impact in terms of discovering even new kingdoms of life, as well as for relative ease. But we all know that 16S is not perfect, but it has had some utility. But, highlighting that point deeper is, for example, the different lineages here you can see, 16S versus just general amino acid identity. AAI can separate out the lineages of E. coli, for example, or Shigella, different species, where 16S makes things to some degree ambiguous from this paper published last year. And so I think it's interesting that really never before in biology, and probably never again, will entire ecosystems and kingdoms be viewed through the lens of one gene. I think rather we're reaching a stage now where shotgun sequencing, or what is often called metagenomics, can now expand the microbiome measurements to query across kingdoms. So as you can see here from this review paper, a little bit of a formatting issue on the bottom there, but you can see shotgun lets you address a lot more and many different kinds of questions than you could from just some of the targeted assays alone. You do have to caveat, though, is that if you're trying to avoid human DNA, you, of course, don't always do that because you're just taking shotgun samples, for example, from skin. You will get a lot more contaminant DNA or host DNA. But nonetheless, I think it's quite intriguing that you really do see a lot of opportunities at this stage with shotgun sequencing. We've published some preliminary work on this and showing that actually you can look at phages that infect the bacteria, sort of the top-ranking phages co-occur with those strains and those species that are most abundant. So you actually have a way to validate what's occurring in your sample, this being from some shotgun city-based metagenomics. But there are, of course, challenges. So there's a lot of excitement. There's a lot of things that you can measure. We're really, the field is changing, but there are a lot of basically challenges on all these methods of prep, extraction, and informatics. So on the prep side and extraction, we've published work that shows a new multi-enzyme cocktail, where they're using, say, an omega kit or the Mobio kit, or doing a various of a hot phenol of different kinds of extraction. For all protocols, we've seen so far that a multi-enzyme cocktail during the extraction gives greater yield of nanograms per microliter. You can see here in red for literally every kit that we've tested. So this has been an ongoing work with the MetaSUB consortium to get better methods for extraction, especially for low input samples. And so this is one of the things we've been focusing a lot on. We've also been looking a lot at what you think for what is present in your sample, especially for things that are potentially pathogenic. And this is one that was notably done in the New York City subway study, where the potential pathogens, even when called by 8 different taxonomic classifiers and metagenomics algorithms, are still not enough in some cases to distinguish between closely related monophyletic strains, for example, some species of bacillus. And so we published a strain level analysis tool, in this case with one codex, that could actually give you an ability to distinguish in situations where you have 99.99% related species, like Anthracis in this case, or other strains of bacillus. You could actually tease them out looking at both the plasmid as well as the specific SNPs, which again would be impossible with 16S. And so using the data and really mining it deeply is a way to give you strain level specificity in a way that we have never had before, and also which is very important for public health as well as for biodefense. This matters actually not just in those contexts, but in other ones as well. So one in particular is in precision metagenomics, and we've seen, again, with shotgun data, you can tease out what's present. In this case, we grew samples that were cultured from environmental media before and after antibiotic treatment with tetracycline, and we could see that which gene was driving resistance for the bacteria on that plate, we could actually pull out from the shotgun data. If you look at the ratio before and after treatment, we could see here for both genes on both media, the coverage of tetracycline K went up much faster. And so this is relevant for work in hospitals, but also, of course, work in even monitoring hospitals with the patients themselves. So that's more in a local context, but we've also been expanding this work into a more global context. In particular, one of my favorite aspects actually of the subway study was this fact that if you take all the shotgun reads and map them to known organisms, really half the DNA matched on no known organism, even with relatively lenient blast criteria, 90% identity across 90% of the length. This was, this number really represents the amount of exciting unknown organisms that are around us that just don't have a good genome assembly and we haven't yet identified. So this is actually not the first time that we've been in this situation where half the world under us is unknown. And this, I would say, is true, of course, when Columbus and Magellan set sail. This was the world as they thought it was when they went out in the world, of course, missing literally half of it. And I would argue that the same thing was true when we had invented the microscope. Suddenly we could visualize this hidden world to us that was literally under our fingertips. And today, sequencers represent molecular microscopes that let us visualize this world. So one of the goals of the standards groups is to collaborate amongst many other groups, and this is a list of a lot of the sites as part of MetaSUB. And New York City is really just one of the smaller cities around the world. So if you go to the website, you can actually see all the raw data and all the work being done by Hub and collaborating cities around the world to build up geospatial maps of metagenomics, antimicrobial resistance markers, and also we're using these data to mine for new biosynthetic gene clusters or new drugs. So this is just a nice visualization you can see if you go to the website as well. So far, we've had almost 6,000 samples taken worldwide and more ongoing in this coming year. This is part of an ongoing effort, not just in our group and also the other collaborating labs, but a global effort really, partially from the Bill and Melinda Gates Foundation as well as with other groups, to map antimicrobial resistance markers and see how they move and their impact in different areas around the world. Also, it's also part of the National Microbiome Initiative. So the last part I want to mention for MetaSUB is we're actually using some of these tools and collection devices to look even at large-scale human migration events, as we just completed the collection at the Rio Olympics to see what happens to a city before, during, and after this large-scale human migration event. And we pair this every year on June 21st with the Global Ocean Sampling Day group to monitor essentially what's happening to the metagenomics of the world's dynamics at least once a year. So this again will be repeated again when we get back to Rio. So that's sort of more normal and city-based and AMR context. As I alluded to, though, there are extreme contexts where we also really want to use better benchmarking DNA standards and tools. And this is also something you can look at the Extreme Microbiome Project if you want to check out the website. But the main reason we really want to be here and that you're here today is to think about, in this universe that we all share of ever-increasing entropy, I like to say that the only thing that can really save you is controls. So in particular, when I say controls, I don't just mean in silico controls where you computationally mix together species. I would make that to be sort of analogous to Plato's forms, or the Platonic forms, where they sort of represent these eternal forms of what if you could perfectly mix together organisms, which of course is to some degree useful, but it's the sort of messy world of actual physically mixed together organisms that are then extracted, prepped, and sequenced, where you get, I think a better view of the sort of conflated amounts of noise that can happen in a laboratory. So in particular, in the past year and a half, we've put together a variety of both simulated and biological data sets, all of which are available on the IMMSA, NHGRI, NCBI website, but these include samples some of which have been published already from Nicholas Sagata and other great papers from other groups, but then also sequence samples of standards that have just recently been developed, I'll talk about today, and other data sets we've put together, again, of both simulated and biological mixtures. So two I want to highlight most notably one is the ATCC controls. This is one set of controls of 20 species they've mixed together. They also have more controls coming out very soon. And also Zymo has a biomics control, which you can see here has these 8 species here, both gram-positive and gram-negative, and they try to look at what would be an unbiased extraction versus other potential suppliers that we know we're not getting perfect extraction. So this is on the extraction side, and these are the physical standards that are available. But I want to focus most of the talk today about some interesting things that we've seen on the tool side, on the algorithm side. And in particular, there's an ongoing question about people say, well, what should I use? Which tool do I use? Here's one short list of all the different kind of computational tools in metagenomics. And it just cannot be that everyone is the best or that everyone has their pros and cons as an ongoing question on many forums. And we've been in the process of finalizing this paper's in review. We just got the reviews back and we're in the middle of putting it back in. But it's to really look at this question of benchmarking a variety of different algorithms. You can see them all listed here, and to see how they do for a variety of different standards data sets. So these are all very different algorithms, and we've actually clustered them by, the resolution here is a little bit low, but you can see here we've clustered them by their, sort of when they came out, what's their heuristic and their method of function to map species, how many types of species and genomes do they have across kingdoms of life that are present in their current databases, do they do strain level or look for antibiotic resistance markers, and some other factors about do they have, say, for example, tutorial or other parameters for the runtime. So we've used these on, as I mentioned, a good total of over 30 data sets that represent over 800 species titrated together. And one of the simplest versions was just this Zymo control, where again you can see the 8 species of bacteria and also the 2 fungi. And we just wanted to detect, here's the theoretical composition, how did we actually do when we sequence them? So if you extract, prep, and sequence, we've done it both with Nextera and Chi-CCAP-X. We can actually see here, in this case, even with different cycles of PCR, using one codex to map the reads, we can actually see we really get some of our expected friends and observed friends with a little bit of a splash of human, which seems to go away a little bit with more PCR. But what's interesting is we took, this sample was relatively simple, had these 10 species. When we take things that are more complicated, other published data sets, so for example, if we use the ones that are known, if we take some other samples that we have that are titrated published data sets that have, say, 53 species that we know are in this sample, what's interesting is if you say, what's the overlap between, say, Megan, Clark, or different tools there's definitely a lot of wings to this Venn diagram. They do not always agree as they should, as you would like. And if you would say, give me only your top 20 species, they actually disagree even a little bit more. And if you say, do the similar experiment, if you say you only have 22 species in this Illumina sequence data set, but you push the algorithms to predict more than that, you can say they very often will, but the disagreement gets much higher as you ask them to predict more things that are actually present in that sample. There's another example here, which is 10 taxa. Again, the truth sets in the middle. If you push them, many of the tools will predict things at low abundance that are not actually there. But one piece of actually interesting and I think good news is that we put here on this axis the percent agreement on average between the tools and what's actually the number of species in that sample. And here is the truth set on the right. So you can actually see the mode of the distribution of agreement between the tools is where the likely actual number of species in this sample is. And so it shows that even though tools will and can and often do disagree, where they do tend to agree the most across at least these 12 tools shows a more likely spot of the true number of species in a sample, which has been often hard to discern. And you can see here the summary between the sort of last peak and truth size is actually quite good for what you'd expect to see. So that's on the prediction side. We've also then wanted to see, well, what's the positive predictive value of, say, pairs of tools coming together, essentially putting them together, not just tools by themselves, but also combinations of different tools. And you can see when you combine them, they very often do much better. And this is also true if you do a 3-tool sample, you put together 3 different tools, really your positive predictive value goes very much higher. And so, but you do lose some sensitivity, which is often expected in this trade-off. And it also, one thing to know is it's not always better with 3 tools, but it's generally not worse. I'd say here you can see some of the data of combining the tools. Overall, we've seen, we're in the process of also noting that really some samples themselves are much harder than others. So not every sample is equally hard for each algorithm. You can see here how we're doing for different summaries of different tools and which samples are doing, with more tools get wrong and more tools get right. You can see here, this is sort of essentially what we see for the average positive predictive value for these different tools. And moreover, if you do a rock plot, a receiver operator characteristic plot, you can see there's other trade-offs for different tools. And these are, the not final versions of these plots. We're doing some updated code for new samples, but you can see here there are some trade-offs there as well. So that's on the detection side. That's one problem. But we've also found that abundance can also be quite difficult. And so if you look here, how well do different tools predict the actual abundance of species, not just the presence? You can see we get a wide range of variation for some algorithms, whereas others can maintain a very tight distribution around the expected abundance for different organisms. And this you can see here is, again, a wide range of variance for different tools, but some samples are harder than others and some tools do better than others. And we can see that this is another factor as well. So this is, again, an ongoing work, not just at the broad level for what do you get from a sample, but also read-by-read analysis is also something we've been looking at. This is work with Rashid on it, is to see how do the algorithms do if you only have 5 reads, how well do you do, not just across all the markers of all the reads. And here we've actually seen that Clark does particularly well as One Codex does quite well, but BLAST is also pretty good here. But you can see that you can get a wide range of specificity and sensitivity for different tools at the sort of read-by-read measurement. And you can see here how some of the tools are performing. And again, these are in review now, and the final results will be posted online actually very shortly. So that's at the measurement and the abundance and the detection side, but there are other ongoing questions that, of course, arise from metagenomics research. So what about DNA modifications or RNA as well? Here we can actually see the epigenetics. When you look at on PacBio sequencing, you can actually predict modified bases from the kinetic information from the PacBio data itself. And here when we've taken the Zymo strains and other pooled samples, we can see that the pooled sample has a different signature than the individual strains for their potential base modifications that are present. And so far we can actually see this class 1 standard does have some really interesting motifs, most of them for methyl 6-adenosine or methyl 4-cytosine, which are expected from some of these samples. But it's always good to remember not just the genetic standards, but also these sequencing these controls on single molecule sequence methods, much like PacBio or Oxford Nanopore, will give you an ability to look at the epigenetic states as well of these controls. And eventually, there will be more examples of direct RNA sequencing. In this paper, this was the first example of using this same idea of a kinetic information for polymerase incorporating bases to monitor the modified status of the template strand. But in this case, we worked with PacBio and Jonas Korlak and others to put together a reverse transcriptase at the bottom of a ZMW to look at the kinetic information, essentially watching cDNA synthesis occur in real time for regular adenosines versus modified adenosines and look at the change in the incorporation speed. So this was the first time that we showed that you could actually detect modified RNAs directly as you observe cDNA synthesis. And this is relevant, of course, because we've seen in the last year that both HIV as well as a lot of other viruses are methylated. The RNA itself contains methylation of methyl-6-adenosine that's present, and it's not just for HIV, it's also dengue, yellow fever, Zika, and West Nile virus. We see that literally every single RNA virus we've looked at to date has slight variations of these peaks in red. We see focal spots of RNA methylation, the significance of which is likely controlling their nuclear export, localization, and also their function during infection. So it's a really exciting time for microbiome, metagenomics, and also on the RNA side, the metatranscriptomics, or as the RNA viruses like to say, it's just their genomics, of course. So the final piece I want to bring up, the importance of standards and where else are these standards taking us, so is the actually interplanetary context. It's really sort of low Earth orbit context, but quite seriously, we are working with NASA on several missions. One is the NASA Twin Study, which is to actually look at the genetic aspects of spaceflight and also to improve diagnostics during spaceflight on the space station. So in particular, this is the work with the Twin Study we're looking at, as you can see here, Circos plot of all the multi-omic data. So this is genome, microbiome, epigenome, transcriptome, epitranscriptome, metabolomics, as well as vasculature cognition, and a real integrated immunological profile of astronauts 6 months before flight, 1 year in space, and 6 months upon returning. This is Mark and Scott Kelly, who generously donated literally blood, sweat, and tears to the study. So this is an ongoing work with NASA to improve this. And so, again, when we're sequencing someone far away in the space station, we want to make sure that we get it right using some of the aforementioned controls. But more excitingly, we also have been able to think about not just bringing all the samples back, but could we actually do sequencing in space rather than have sort of the hassle of things burning up in the atmosphere sometimes or taking a while to get back to you. So this is an ongoing project led by Aaron Burton at NASA. It's called the Biomolecule Sequencer. We're working with them to actually do diagnostics in space. So this is so as always with a mission, you need a patch. So you can see here is the mission patch for the Biomolecule Sequencer project. You can see here the nanopore looks like a little rocket going into space. And as we were actually planning this mission, we were working, and I was chatting with Andy Feinberg, one of the NASA Twins PIs. We were talking about starting to send one of these up, and as that happened, we actually had supplies going up to the space station, and the CRS-7 actually blew up on the way there, which was very sad because it's never good when your lab supplies explode. But my favorite thing about this explosion was a really touching letter I'd gotten from NASA that said, really encouraged us to hang in there and keep going, even though they blew up our stuff on accident. So we're really going to maintain the course and keep fighting for the science. So it was a really touching letter. I'll note that when you get a cut from the NIH for your budget, you kind of just get an email. I think this is a little bit more personal. I encourage the NIH to do the same. So, and also space is hard, as Scott Kelly tweeted. So we did have an opportunity, though, as we're planning the mission to, and as we're waiting for a resupply rocket, we thought, well, let's actually try it on the parabolic flight simulator, because in this case, you get a little bit of zero gravity, but only for about 12 to 15 seconds. But let's see if it'll, maybe it'll work there. So in this case, it was Andy Feinberg who actually got up there and did manage to load a flow cell in zero gravity with these positive displacement pipettes, got it up there working, loaded the flow cell, and we actually showed about a year and a half ago that it was possible to sequence in zero gravity. And the interesting thing is we observed some slight fluctuations in current that we now see are just part of normal flow cell variation. But at the time, we were very intrigued by this concept that the flight current data looks like it was different from the ground data. The exact same library done on Earth as well as in zero gravity seemed to be shifted. And again, this is now more just normal flow cell variability in terms of the picoamp current of essentially what's going through the pore per kmer ordered by HMM, which is now an RNN. But we can actually see here that this gives you sort of a waveform, and it's kind of inspired us to think about, well, what are other places where we see waveforms, and can we use them to match to basically different databases? One particular one is in Shazam, of course, and so we had a lot of conversation with Edward Oakley about this idea and thought about, well, let's actually see if we could create an algorithm that would do this to match to known waveforms, which we called UNFOG, the open source Song of Genomes algorithm, to actually match the fingerprints of genomes. And that's what you can see here. And this work was recently published in the journal, Nature Partner Journal Microgravity. So you can see here that in 0G, the current versus 1.8G, and then 0G, 1.8, 0. As you go up and down the flight simulator, you can see the impact of gravity on the nanopore is actually not that bad at all. So it's good to see that the pore works great in 0 or a higher gravity as well. And that it'll work when you get to the space station was what we thought would happen. So As we're continuing to plan the mission, this is actually Kate Rubens, who's getting ready to go up to space and loading her first flow cell, doing planning missions at Houston. She eventually got up to the space station no problem, and actually when they sent more resupplies up there on the CRS-9, you can see her the perfect booster return and landed back down Earth. And as of last August, we can see that we can sequence in space with relative ease, actually, and that Kate Rubens was the first person to sequence DNA off this planet, going up to about 1 billion sequence reads, and this work is now in review as well. But one notable piece of the data was actually that we could see, if you look down here, that the percent identity for 1D and 2D reads, this is older chemistry, the version 7 flow cells, was pretty good, and actually was slightly better for the flight data than the ground data. So it seems like the zero gravity sequencing really gave no problems at all for the device and for the technology to work well. So we were able to take all these reads and put them together to do a de novo assembly of E. coli. And as you can see here, compared to the gold standard PacBio genome assembly that we did with the same sample here at Cornell, we can see we're able to get almost perfect genome assembly when compared to PacBio. And really, we can demonstrate that this is the first genome sequenced and assembled from data not made on this planet. So it was still orbiting the planet, mind you, but not from this one. So it was a pretty exciting set of data to get over the past 6 months and put together. And I can tell you also, we've been getting additional data from the space station, doing additional replicate runs. And the data looks very, very good. So this actually means that the flow cells last likely long enough to get to Mars. If you wanted to send something to Mars to try and do a diagnostic there, it looks like they would make it. So it's pretty exciting. And also, we are sending up the Zymo controls of the titrated mixture. Will be a truly, a control used for all contexts, including space. It just is flying up there in the next resupply mission, and we'll get that data back down to Earth very soon. So this is essentially what the new poster actually for NASA is. They call it sort of the Space Genomics poster. I actually have one of those here in my office right now. You can see right there that they just sent me today. So this is the idea that we really can use sort of integrated molecular diagnostics and profiling and personal omics profiling to really look at all the dynamics of the human genome, the human microbiome, and really the total sort of biome that is the space station and sort of the humans on their way towards other planets you can see here floating towards Mars. So it's a pretty exciting time for molecular diagnostics, both here and on other worlds as well. So I want to also point out that, I think it's not just thinking about how do you measure things, but eventually I think the real test of understanding will be how you also grow things. And so this is part of the Bioacademy class that we're also working with some of the people in the Church Lab on this and others. And I also want to thank finally people from the Brooklyn Bioreactor, which has given us some of the most challenging, mysterious, and peculiar samples to look at for some of the standards development and computational methods testing. And as you can see here is the plan here is to do continual microbiome monitoring with the Brooklyn Bioreactor. And along the way, we will be using the standards, both physical and computational. And really the next steps I'd say is that, more kingdoms is harder, but I think that's better. I think more data is more happiness. We use this positive control for all the MetaSUB site collections and have been using it in the clinical lab as well. This really is our true positive control. We use both the Zymo and the ATCC as well as the ABRF MG RG control. So we use them all, and this I think points to the fact that we need really a constant gardening effort really as a community for both the tool standards and the benchmarking to make sure we all improve things together and use the standards so the data is comparable between sites. And of course that's not really new. You should always have positive and negative controls. So at the end, I want to thank everyone from the laboratory who really helped with this project, and really it wouldn't be possible without them. So deep gratitude to the funders, including the World Fund Foundation, the NIH, the Bailey, as well as the NHGRI, and also the Pershing Foundation, also Gates and Sloan Foundation. Thank you very much for your time, and have a great day. I'm happy to take any questions.
Thank you for that wonderful presentation, Dr. Mason. We want to get right to the questions and input. So here's a reminder as to how audience members can communicate with us. Questions can be submitted via Q&A button at that lower left. If we are unable to get to your questions due to time constraints, Dr. Mason will be answering them submitted questions via email. So let's look at our first question. Dr. Mason, what algorithms are best for bacteria or viruses?
Great question. So different tools have different tools have different databases, as was we pointed out in one of the earlier slides. And that's really one of the biggest factors in what you'll detect is what's in the database of the tool that you use. So we've seen, though, that really most of the tools that have bigger bacterial databases do a really good job. So this includes One Codex, Cosmos ID, Blast Against Everything. Metafline does a pretty good job. It's recently expanded its databases, and also Clark has a very large database. So those are all those tools are some of our go-to tools that we use pretty routinely. And so those are the tools I recommend. But the biggest factor is the size of the database, both for the viruses and the bacteria.
Thank you. We also have another question from an audience member who wants to know, are DNA standards, can they be purchased today?
Yeah, good question. So they're actually publicly available. The standards from both Zymo as well as ATCC and the MGRG are also synthesized by ATCC. So all the standards I've mentioned, the physical standards, the ones you can use in your lab, are available both as pure DNA as well as pure lyophilized cell extract. So you can get them, use them, test them, and really get them going in your lab. You want to make sure the standards are usable long-term and for many experiments. And so that's you go ahead and hop online and get them. And if you have any questions or you can't find them by Google, you can send me an email.
Thank you, Dr. Mason, again for your wonderful presentation. Do you have any final comments that you'd like to make for the audience before we end?
I guess I'd just say everyone to stay curious and know that we've really just started discovering all the world that's already around us. So I think it'll be a very fun period in science for the next 5 to 10 years that we can really suddenly see this invisible world. So, but that I would encourage everyone, while taking up that excitement, to don't forget to always have, like you do on every plate in lab, a positive and a negative control, and make sure you've got some sanity checks, and then you can actually have a lot more faith in interpreting your data and discover more things.
Wonderful. Thanks again, Dr. Mason. I'd also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available for on-demand viewing for up to 6 months. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We encourage you to forward that announcement to your colleagues who may have missed today's event. Thanks for joining us today, and we hope to see you next time. Goodbye.